In this video, let me start from a cell. Okay, so what is a cell here? So all living beings are made up of cells. So all living beings means here it includes plants and animals both. Okay, now all the living beings here, living beings, everything it is made up of what? Cells here. So you might wonder what cells actually look like. Okay, so how it looks like now. Let us take a closer look at the basic structure of a cell using a microscope here. As we know that cell is a very tiny thing. Okay, so with the help of microscope only we can see that. Okay, now with the help of microscope, what we will do, we will see the structure of the cells here. Okay, to see the structure of cell, we have to do one activity here, activity 2.2. Let us study a cell here. Now take an onion bulb from your kitchen or garden, wash it thoroughly with the water here, like this of a onion bulb. Okay, now cut an onion bulb vertically into pieces. We have to take a piece, separately piece we have to make like this. Okay, now take a one piece of onion and pull out a thin transparent layer from its inner surface with the help of a forceps. So with the help of forceps, this is the water thing. With the help of that, what we have to do inside, inner layer of this onion, that we have to take a one layer. So how it is this? This is a very thin and transparent. Okay, now place the peel in the petri dish. This is the what petri dish we will call. This is a petri dish. It is completely made up of what glass here. Okay, so place the peel. Which peel? This is only transparent thin peel from the onion of the inner surface of the onion here. Okay, now place the peel in this petri dish here containing a few drops of saffron in. Saffron is nothing but what here? It is a red color stain here. For the 30 seconds we need to place in that. So this will give a pinkish color to the cells. Okay, so due to this color, the cells, it will become a pinkish in color. Now which is help us to see them very clearly here. Okay, now with the help of thin brush, transfer the onion peel to the another petri dish here. Okay, which is containing water to rinse the peel and remove the extra stain here. Okay, so that's why we need to transfer to the another petri dish here. Okay, with the water, what we have to do, we have to rinse out. Why? Because extra whatever the color it is there, no red color, this is stain, it will go now. Okay, now put a drop of glycerin over the onion peel here. Now on the slide, this is our slide. Again we have to transfer from to this one, and on this we have to put a glycerin here. So why we are adding here glycerin? Because the glycerin will prevent drying of the cells here. So the cells, it will not get dry here. Why? Because we have removed this thin layer. Okay, so what will happen? It will get a dry. So to avoid that, what we are adding here glycerin. Okay, so what is the function of glycerin here? Adding here that it will prevent the drying of the cells here. Very important. Now improve clarity for the better visualization of the cells also. Not only it prevents the drying, it also improve the clarity of the cells visualization when we'll be seeing. Okay, now slowly place a cover slip over the peel using a needle here. Now here it is the slide. Okay, on that what we have to do, with the help of this needle, we have to place one glass, and that we will say that is over that, such that, okay, slowly cover slip we will say. Okay, cover slip. Okay, over the peel using a needle here, such that no air bubble get trapped here. So there should not contain any air drop after closing that, like this. Okay, now use a blotting paper to gentle, and we have to just wipe the extra if it is anything. Okay, we have to just remove the edge of this cover slip here. Okay, now weave the slide under the microscope or a fold scope here. Compare it with the figure 2.3. see here it has been shown here. Now this is how looks like in the microscope, the cells of the onion peel here, and these are what cells of the onion here. Now see here, this is outer, what it is there, no outer side, that is a cell wall, and the next to that, inside, which is a cell membrane here, and the round whatever you can see, that is a nucleus. Cytoplasm is nothing but which is present between the cell membrane and nucleus here. Okay, now this, see here, next what they are saying here, what similarities do you find in the figure 2.3 C and D? Is it looks like a brick? Yes, it's looks like a brick here. Okay, now you will observe nearly rectangle, not exactly, nearly it looks like a rectangular in a shape, okay, or structure under the microscope. Okay, now these are the cells of the onion peel here. which are closely arranged, without any space between them, which is very closely arranged here. There is no space between the cells here. Okay, now try to observe the peel of the leaves of any other different plants around you. You will find that the plants are made up of cells, not only onion. If you are trying with any other plants, all the plants also, it will comes to know that it is made up of what? Cells here. Okay, now what do you think the body of the animals is made up of? The next question is what? About the animal here. Okay, so to understand that, we are going to perform another activity, that is 2.3. Let us investigate here. So rinse your mouth with the clean water here. We have to rinse our mouth with the clean water. Now use the blunt end of the clean toothpick here. I'll show the picture here for the blunt end of the clean toothpick. So here, this side, this is what the blunt of the toothpick here. Okay, now after that, use the blunt end of the toothpick and gently scrape the inside of your cheek. I'll show the picture for this, like this, inside the cheek here. Okay, this one, this part. Now after this, place the scraped material in the drop of water on the again clean glass slide and spread it evenly. Where we have to place? We have to place in the glass slide, and the glass side, it should contain already drop of water in that. And after placing, we have to spread evenly. Okay, now add a drop of a methylene blue. So in the onion peel, we have used a red color. Here we are using methylene blue color, which is a blue colored stain over the material on the slide. Adding a stain again improve the visibility of the material under the microscope by increasing the contrast here. So basically, why we are using the stains here? Because it will improve the visibility of the material under the microscope by increasing the contrast here. Very important. They may ask you for the 1 marks here. Okay, now after adding this blue color, just leave it for 1 minute. Add a drop of again glycerin, as I told you in the last. Why we are adding glycerin? It should not get a dry here. Okay, now carefully place a clean cover slip like in the onion peel on the material, and the remove excess glycerin from the edges of the cover slip using the blotting paper here. Okay, now observe the slice under the microscope and draw what you see in your notebook here. Now see here how we will see. What do you observe? You will observe a polygon shaped structure as shown in the figure 2.4. This is how we will start looking like this. Okay, it is which is in the polygon structure here. Now these are cheek cells. These are the which cells? Cheek cells, which form the inner lining of the mouth. Okay, so these cheek cells, what it is forming? Which is forming the inner lining of your mouth here. Now what similarities and difference did you observe between the cells of onion peel, which have done in the activity 2.2, and the human cheek cells in the activity 2.3? So now we have to find the similarities, what and all the similarities between these two cells, and what are the difference we will find between these two cells here. Okay, now you have observed that the cells have three main parts of the thin outer lining, central region, and the small round structure inside it. So from this both activity, what we observed actually in the cells which is having three main parts. One is thin outer lining, okay, like how we are having a skin like that. Okay, now central region, this is what we can say complete. This is what central region this is. Okay, I can between that, what it is having? A small round structure inside it. This is round structure. This also we have found in the onion peel also. Now structuring. The outer layer is, we will call, this is cell membrane. This outer layer we will call cell membrane. The round structure in the middle, this round structure, this is what? Middle, which we will call nucleus. Okay, which is also covered by a thin membrane. Okay, so this nucleus, what it is then, which is also covered by a thin membrane. Okay, the space between the cell membrane and the nucleus means the space between this is what we will say. This is the cell membrane, this is what nucleus. In between the space, whatever it is there, no, that is completely filled with the cytoplasm. Okay, so these three I mean cell membrane, cytoplasm and nucleus are the basic parts of the cell here. Some cells like onion peel cells have an extra outer layer, that only we have said in that cell wall. Okay, let me show you the picture. So in this it was extra, that is cell wall. Okay, otherwise it is having a membrane, it is having cell membrane, nucleus and cytoplasm. It is also having here in the onion peel. But otherwise these are the main three basic parts will be in the cells. What is the importance of these structure in a cell here? Now what function do they perform? What are these functions important for the maintenance of the life? So these three questions are very important here. Okay, so as we have discussed, cell, it is having three main parts, which are cell membrane, nucleus and cytoplasma. Okay, now what is the importance of a structure of the cell here and the function of what they function they perform, everything we are going to understand in the next one. Okay, now the cell membrane encloses the cytoplasma and nucleuses. It is like a it is enclosed the it is closed the completely cytoplasm and nucleus. Okay, the like it is acting like a what here, wall here. Okay, now the cell membrane separates one cell from the another cell also. Okay, like this you can see here, this is one cell and this is one cell. How it is separating? Or separating by a cell membrane here. Okay, from another, it is porous and allow the entry of a material essential for a life process and the exist of a waste material. So here it is means which one here, cell membrane. Cell membrane, it is having porous means it will be having kind of a holes here. So what will happen through that, what will liquid or air it will get passes through that. Okay, so it is having a porous and allows the entry of a material essential for the life of process. For which life? For the cell life. Okay, so whatever it is required for it, no, that material it will get enter through the porous. Okay, porous where we will find it in the cell membrane here. Okay, so which helps to life process, and the exit also it will get a comes out through the porous here, that is waste material, because this cell doesn't have a kind of digestion system, excretory system what we are having like that. Okay, so everything which happens through the cell membrane here, because cell membrane which is porous. Okay, now we will go to the next one. So if they ask you what is the function of cell membrane, we have to write this one. Okay, the next comes, that is cytoplasma. So where is the cytoplasma present in actually cell, which is present in between the nucleus and membrane. This is what everything which is cytoplasma only then. Okay, so cytoplasma contains other components of the cell. Okay, for the cell whatever it is required, no, some other components also which is present in the cytoplasma here. And the compounds such as, which compounds are present which is present in the cytoplasma, which are carbohydrates, proteins, fats and mineral salt here. Okay, so most of the life process takes place within the actually cytoplasma here. It's very important. Okay, so this is what the function of the cytoplasma here. Now we will go to the next one, that is nucleus. Okay, the nucleus regulates all the activities that occur within the cell here. So whatever the activities which is happening in the cell, no, who is regulating? The nucleus which is regulating, and it also regulates the growth. Okay, now this is the function of the nucleus here. Okay, the cell wall in the plant, this cell wall which is not present in the this cheek, okay, inner lining of the cheek, but which is present in the plant. Okay, so here now we will see what is the function of a cell wall here. The cell wall in the plant cells provides the rigidity and strength to the plants here. Cell wall, what it is providing in the plants, it is providing the rigidity. Rigidity is like a stiff. Okay, it will be very difficult to bend that. Okay, so that it is providing cell wall here, and it gives the strength to the plants also. This is why all cells are what, which is arranged in a compactly, means which is arranged very tightly and look firm in a structure. Firm is nothing but what here, it will able to stay in the same shape. It is not going to change the shape or when we will press or whatever. Okay, so it will going to remain as it is. That's why it looks up firm in the structure. So what who is doing all this work? This is with the help of cell only. Cell wall only it can be possible here. Okay, now in the next video we are going to study about the a step further. Here we are going to find the difference between the, here it is an animal cell and here it is the plant cell here. Okay, thank you.